Good evening, everyone, from a very warm Stellenbosch and Cape Town. I see some participants are joining up already, and Professor Stephen Jeffrey is actually going to join us tonight as well, but he is running a little bit late. So before we kick off with today's topic, which will be neuropsychiatric genetics, I just wanted to address one of our viewers emailed me about a topic that we had a few weeks ago about genetic screenings, and we spoke about PGT and NIPT. Now, they asked if PGT will be able to replace NIPT, and just to clarify, PGT cannot as of yet replace NIPT completely, as PGT is only used in vitro fertilization cases. The P in PGT stands for pre-implantation. But that out of the way, tonight is all about neuropsychiatric genetics in a way that makes sense in the clinical room. So it's not necessarily a case of this gene equals this disorder, schizophrenia or depression or bipolar disorder, whatever neuropsychiatric disorder you might be talking about, but it's all about how genetic variation influences brain development, brain regulation, and brain circuits. We'll talk a lot about regulation and circuits and systems in this webinar tonight. And we'll also talk about what changes how we look at these type of things and how that changes how we design the care for this for neuropsychiatric disorders. So there are two ground rules, I would say, or two ground things to remember for the topic tonight. First of all, genetics is not destiny. All right. So in psychiatry as well, psychiatrists already grasp this concept that a gene shift is only probability, right? It's a risk factor. It's not certainty and it's not causation. So, for instance, if you have, especially in the case of polygenic disorders, which we'll talk a little bit about later, if you have all the risk variants, that doesn't mean you will get the disorder, but it does put you at a higher or a lower risk. The second ground rule or important thing to say is that these webinars are purely for education. So it's not personal medical advice. I will focus on the clinician mental models and real published examples and try to adhere to the science as best I can, but this is not medical advice, this is purely educational. So let's start off by defining what we mean by genetics in neuropsychiatry, because people can be referring to different things. I would say there are three levels at which you can look at this type of thing. So the first being rare variants, or high impact variants. So we spoke quite a bit about that in that same webinar about the screening tests. And these, we'll go into detail about the three in a second. So level one is rare variants and high impact variants. Level two would be common small effect variants. Those are the polygenic ones that I referred to just now. And then the other level would be gene regulation. So that's how genes are turned on or off or up or down or enhanced. And also at specific times in specific cells. And for tonight's neuropsychiatric focus, it will most likely be in some form of brain cell or some anatomical part of the brain. So when clinicians hear the word genetics, we often picture single gene disorders. And that is sometimes true. You get neurodevelopmental syndromes that have one specific gene or gene sequence or gene polymorphism that directly links to a disorder. But especially when we're talking about neuropsychiatric genetics, it's almost always on a spectrum, right? It's not as binary as on or off. So in those cases where it is as binary as on or off, you call those rare high impact variants. These are uncommon, but when they are present, they strongly shape the neurodevelopmental and psychiatric risk. As opposed to those, you get polygenic risk. So these are thousands of usually pretty common DNA variants or differences, each contributing a tiny little amount to a meaningful shift in the risk to develop a disorder or a disease. You also get gene regulation. So this is quite a big one. So this is when you look at many, many risk variants, and they don't necessarily cause a protein to be absent or break or make the wrong protein, but they change how much of a protein is expressed, where it's made, and when it's made, right? How much, where, and when. And that all ties around gene regulation. So to define those terms simply, a variant, so to speak, is a DNA spelling difference. So if you think about the genetic code ATGC, that would be if there's supposed to be an A, you get a T or a C or a G, right? One of the letters, the base pairs, nucleotides are different. That's a variant. And specifically, that's a SNP, a single nucleotide polymorphism. The other term that's very important to know is just risk when we're talking about gene risk. So that's the probability of a shift. It's not a guarantee. So that always needs to be emphasized that these type of things in neuropsychiatric genetics, they're always referring to causations, not, I'm sorry, correct that they're always referring to correlations, not causations. And yeah, and then the regulation, the term that I just mentioned is the sort of, you can think about it almost like a volume control type of thing for genes. So it's a spectrum of more expressed than it should be, or less expressed than it should be, or expressed in the wrong place or at the wrong time. That's the gene regulation. So how do we see this gene regulation in the brain, for instance? So one molecule that's very close to my heart, because it's something that I focused quite a lot on back in my master's, is microRNA, which is one of the most, if not the most important dimmer switch in biology. So in previous webinars, we spoke a lot about the central dogma, which is that the whole thing of DNA making RNA, making protein, that sort of 1, 2, 3 stepwise process. Now, microRNA shouldn't be confused with messenger RNA, because what microRNA is, it's a small RNA molecule that binds to the messenger RNA and either degrades it or silences it. And what this results in is that those messenger RNAs don't make it to the ribosomes in the cytoplasm, and therefore this regulates how much of that protein can be expressed or synthesized. So a microRNA, just to repeat that, is a very small RNA molecule, different, distinguished from the messenger RNA, and it helps regulate gene expression. It's kind of like a post-it note or a stick-on message onto the messenger RNA that says, don't make this protein. So clinically, why does this matter? It's because a lot of psychiatric genetics is all about regulatory control. So it's not one broken gene, but a network of genes being nudged to essentially give you this disorder. And that's the concept of microRNA. And it's not just theoretical. There are quite a few different clinical studies that have looked at the correlation between certain microRNAs and how, in, say, the human prefrontal cortex, this has related to different disorders. So in 2024, for example, there was a study that looked at the brain region mapping microRNA in the dorsolateral prefrontal cortex, which is a region of the brain that's heavily involved in executive function and working memory and cognitive control. And they looked at microRNA expression in this area. So just before I delve a little bit into that study, there are 3 definitions that would be quite important to know. The one is a quantitative trait locus, which is, so QTL, that's a DNA region associated with a measurable shift. Then you get a specific version of that called a microRNA QTL. That's a DNA variant that shifts the regulation, the amount of a gene being expressed. And then you get the term called GWAS, Genome-Wide Association Study. And all that basically is, it's a type of study that looks at a bunch of genomes across a population of people. So your sample size is a lot of people, and you're looking at their entire genome. A genome is all of the genes of expression in a person. And I see we are joined by Professor Stephen Jeffrey. Stephen, how are you?
Hi. Sorry, because I'm a bit late. I had some clinical commitments. So, yeah. So
No
I
problem.
feel like I've missed probably one of the most important parts of the whole thing. But yeah, I mean, just to step back, I think from us clinicians, this is the very exciting part of what we talk about on the genetics webinars, and what Blue Rose will ultimately have one of its main focuses. I know this is I don't know if you just your background, Chris, in terms of your research focus on the neuropsychiatry and the whole neuro side of the genetics. Was it something that you focused on in the beginning?
Just in passing.
Okay, but I mean, I think Chris needs to just let us know that this is something that was a very important part of your background, your master's. What was the focus of your master's thesis, Chris?
So I looked at quite a few factors. I looked at microRNA and how that in a African context or the African admixed populations that can relate to schizophrenia medication, so antipsychotics being leading to adverse drug effects or yeah, that type of thing. So in a nutshell, to phrase it simply, is looking at drugs, the environment, and microRNA in a sort of epigenetic lens. And yeah, and that is a big thing. And yeah, so this is very much I love the brain and I love genetics, and so this is a topic that I'm extremely passionate about. Yeah, and this is what my focus has been on for quite a few years.
So in terms of, I mean, I don't know, we and I've interrupted you very rudely. Did you want to just continue with where you were sort of heading, and we can continue after that with the conversation?
Yeah, no problem at all. So let me just catch you up, actually, Stephen. So the we were just speaking a little bit about how there in the clinical setting there are essentially three different types of things that psychiatry or neuropsychiatric genetics can refer to. So just quickly, you get rare variants. So these are ones we spoke about in the screening as well. So one gene causes a disease or a disorder. You get the second level, which is polygenic. So that's many small contributing factors. And then you get a third type of thing, which is gene regulation. So that's not turning a gene on or off. That's controlling how much is expressed or how little is expressed, which cell type it's expressed in, and what triggers its expression as well. So those are just the basic terms. And then I was speaking a little bit about a study, or started speaking about a study that was done in 2024. And essentially what they did, in plain English, they took DNA variants and looked at microRNA levels. So there's a term called microRNA QTL, which is microRNA quantitative trait locus. And what they were trying to see is to see whether these gene regions signal overlap with schizophrenia associated genetics from genome-wide association studies. So in plain language, they were seeing, does the level of this molecule overlap with the gene signal seen from other genes that we know are associated with schizophrenia? And they did. They did identify over 400 of these microRNAs, and a very large amount of them was also almost directly proportionally associated with the overlapping schizophrenia genes. And so that overlap is important because what it tells us is that regulation is coherent. Right? So the same DNA variants that alter microRNA levels also frequently sit in regions that alter gene expression in the brain tissue. So the one controls the other. So what this means is that they used schizophrenia genetics as the disease model, and they mapped these risk variants onto these microRNA. And what they found is this is a correlation that we can strongly see phenotypically as well in the presentation of the disorder. So, and what you draw out of that from a clinical translation is it doesn't mean that these microRNA causes schizophrenia, but it does mean that some schizophrenia genetic risk does appear to be shifting the microRNA regulation, in the specific study's case in the prefrontal cortex, which does have this cascade of effects that is linked to the onset of the disorder. And so the take-home message from that study would essentially be, in a psychiatric illness, if it's schizophrenia or one of the other ones, a genetic risk is often acts through a whole regulatory network of, like, RNA linking to messenger RNA, linking to gene expression. So it's not one gene causing one disease. It's one thing that regulates another thing that changes how much of a thing is described. So it's this whole interacting pattern, and then you can bring things like environmental factors into that, and things that you wouldn't necessarily think are important environmental factors. So you asked about my master's. A big co-variable that I looked at in my master's was childhood trauma, and you'd be amazed at how big of an effect childhood trauma epigenetically can have on adverse drug risk in adulthood. So it's interesting. So your drug metabolism gets affected by environmental factors in early childhood, as a strange example.
And how did you
So
quantify that? So can I just need to ask you? So that was a
yeah,
very interesting finding, I'm sure, and it's interesting to me as well that you actually looked at that, and that was one of the
yeah.
variables you brought into the whole thing. How did you, or how's that childhood trauma quantified, and how
So
did
there's
that
a childhood
actually?
trauma questionnaire. So it's a survey. It's a survey-based response, and so a psychiatrist will have their patient cohort, and they'll essentially be filling in the cohort. Um, and then you would, in that specific case, you would also, another cofactor that you would be looking at is something called the PAN score. So that's for schizophrenia specifically. It's called the positive and negative syndrome scale. And that also looks at the various different domains of the psychiatric illness. So all psychiatric illnesses have these sort of categorical uh, symptoms that you can quantify, and they all have their own questionnaires essentially that relate to, if you score this, then it means it's autism spectrum disorder or it's bipolar disorder. So they do have, or they do attempt to firstly categorize and then quantify something that you could use as a rule of thumb to try and diagnose this. But there is, and the manual that they use to do all of this is called the DSM-5, is one of the newer ones. But there is a lot of conflicting things that revolve around that. And if it was as easy as being able to just check things off a box and diagnose, then there wouldn't be a need for psychiatrists. People could just read a book. So, so there
Yeah.
is, and that's yeah, yeah.
That is the great thing about psychiatrists is that they've remarkable at classifying and giving criteria, and that's why you have the DSM, the Diagnostic and Statistical Manual, in psychiatry, and absolutely important that they bring in these criteria, and I think the marriage between what they do and what you guys are now able to bring in from a molecular level is quite remarkable. Do you get the
Yeah.
feeling that the neuropsychiatrists are a little bit further down the line than the other medical specialties in terms of looking at genetics and their role in the impact on disease, or is that not as?
Factually, I can't say yes or no, but intuitively, I want to say yes because there's even in my even in my experience with working with doctors, I think because psychiatrists do have a lot of sort of I don't want to say guesswork, but a lot of things that don't have easily measurable biomarkers. They can't draw blood and say this is bipolar disorder. That they were really more open to looking at anything that could act as a biological way to measure this type of thing. Whereas a lot of the other medical professions already have their diagnostic tests and are a little bit less open sometimes to do the new things because they already have something that works okay. So yeah, so interestingly enough, from my perspective, the neuropsychiatrists or psychiatrists were one of the first clinical fields to adopt genetic screening in terms of prescribing drug medications and that type of thing. Um, the oncologists also have yeah, there's a lot of oncology um, that's also looking at gene testing and even gene therapy and that type of thing. So credit where credit is due. But I would say psychiatry and oncology are the two medical professions that really do integrate genetics into the and have for a very long time integrated genetics into their fields.
And is is it something now becoming part of routine clinical medicine in any places where they actually look at genetics, or
You know,
is this
I
experimental?
yeah. So I often get told that there are these places, and I often read studies where this one clinic or this one place is implementing genetic tests, and you always read about how successful it was. But here in South Africa, when I go to hospitals and I speak to the actual doctors, they very rarely are integrating. And if they do, it's sort of older technology. It's not always the cutting edge stuff. So it's the difference of what you read in on papers of what is available versus what you see in practice is quite a dissonance, because you don't really see people integrating genetic tests and these type of things into their practice. Do you come across a lot of doctors that actively say they integrate genetics?
No, no, not at all. I mean, that's the whole point of these discussions, these conversations. It's not part of my world at all, to be honest.
Yeah.
And I think that's where we are now looking at a new paradigm in medicine, where as you know, I mean, I practice in a very surgical field, and the whole world of pelvic floor dysfunction, incontinence, as I think a very strong genetic component to it. But and I believe it will ultimately become part of our day-to-day screening for patients, and dictating and advising patients on how to do maybe childbirth, because the genetics will play a role there. We know that.
Yeah,
But yeah, to come back
absolutely.
to genetics, in psychiatry, um, I think just having seen, just having a very mild and very short and more short exposure to what you guys have been doing, I think that intersection between the pharmacogenomics and the neurogenomics, I think is a very strong one. And I think if you, even if you're not a psychiatrist, you also understand the fact that the big challenge in modern psychiatry is the medical therapies, selecting medical therapies, and certainly in Cape Town, patients are on polypharmacy when it comes to psychiatric medications. My patients, I see them, and anyone with a significant psychiatric diagnosis will often be on a broad array of medications. And the question is, can those be better prescribed, and can they be better tailored to the patient's specific condition? And I believe they can. Is
Yeah,
there anything that you want to contribute from that side, Chris, in terms of how
yeah.
we can select the medications and where the genetics come into that?
Yeah, so absolutely. So in also in 2024 there was another paper that where they called it the caring, so caring clinic. Now caring was clearly an acronym for something. I unfortunately don't know what it stood for, but what they did in that sort of hub and spoke mental health professional model, they implemented some genetics, and it was around 300 patients that they looked at, and they implemented this genetic testing, and what they found was that essentially almost half of them had a pathogenic or likely pathogenic genetic contribution to their disorder. And even if the even if the actual uh, treatment or diagnosis didn't change, what often did change is they either incorporate, when they found, okay, this does have a genetic underpinning, they did cascade testing. So they did family testing, seeing if some of the family members are affected. They did family counseling as a consequence of that. Sometimes the medication type did change. So the diagnosis didn't change. They still have this thing, but they realized through genetic analysis and genetic testing that this is the disorder that you have or disease that you have, but this is not the best medication for your specific genetic profile. So they changed the medication. Sometimes, if in enigma cases where they couldn't figure it out, they actually referred some of those patients to clinical trials to go figure out what is going on here. Some of the patients they gave additional medical surveillance because they found additional risk factors. They did further speciality referrals, so they found out what I thought had been a psychiatric disorder was actually a rare genetic disease, and therefore I had to get a different speciality involved because this was actually a sucking an example out of my thumb, a brain cancer, or it was not what I thought it was. And more than half of them, I almost want to say more than 60% of them had a dosage change. So there was, yeah, either they were given a less dose or a higher dose of the medication that they were already taking. So this is when the and this is one of those examples where this caring clinic was doing this, where they were implementing this in their study, but since then, I haven't exactly seen a bunch of clinics looking at the results and going, well, that's a no-brainer that this is something that needs to be included in the clinic. Um, yeah, again, that study actually, come to think of it, highlighted a very interesting thing that happened or happens when you look at rare diseases. So after that study was published, there was another one about a year later in the European Journal of Medical Genetics, where they looked at people with rare diseases, rare genetic diseases, and they specifically looked at people with neuropsychiatric symptoms, and they looked at something called diagnostic delay. So you know what diagnostic delay is, right? So what they found is that there was an average diagnostic delay of almost 6 years, right? And what they also found is that a lot of times when people had neuropsychiatric symptoms, so let's say psychosis as an example, this started showing on average almost around 4 years before the actual diagnosis was made, right? So what this means is that sometimes people see neuropsychiatric disorders and they report this is burnout or PTSD or psychosomatic or depression or ADHD, PTSD, schizophrenia, autism spectrum disorder. And then what they've essentially realized is that this was a complete misdiagnosis and that the psychiatric or presentation of it was a symptom essentially of a rare genetic disorder. Um, And what they then did as a follow-up, they after diagnosis and disease-specific treatments, they changed the treatments and they were able to actually treat the real underlying cause of what was going on and change the medication, um, and yeah, and actually improve the care in terms of psychiatrics.
And that was more of a, is that an experimental clinic, or is that something that is running now as a day-to-day standard clinic where people will go to?
So no, so that was a single study. So there was one cohort of yeah, it was around 300 people, and I don't know if they after the results whether they yeah, as you know, with a lot of times with these things they find these results and then nothing nothing comes of it. They realize it does what we're doing doesn't work, and then they go back to doing what doesn't work. So, and it's something
Uh,
I don't
um,
yeah
yeah,
yeah
I'm interested in the fact that these things are out there. We know they work. The results from that study were remarkable,
yeah
and yet, as in many parts of medicine, the rolling out into clinical practice is slow.
yeah
And where do you think the real sort of bottleneck is, or the blockage is in terms of rolling this out into day-to-day medicine? Because clearly there's something there, and maybe more
yeah
research needs to be done. But in the end of the day, I mean, patients can be helped through this, and there's very little harm done in getting the genetic knowledge.
yeah
And
it's
that's the
yeah
point. You're not, it's not like you're going to do an operation to diagnose something and then, but I mean, you're absolutely doing something that is accessible and, harm, and first doing no harm. So, why don't we do more of this?
yeah no it's a great question, and there's always this push in medicine for non-invasive alternatives to treatments, right? And it's interesting to me how there is often this push for a drug-based application as opposed to surgical-based. And genetics seems to me that when we have a result that does deliver a better diagnostic criteria, why are we not using this? And so there's all sorts of things that usually get mentioned in these conversations, like the cost of implementing these tests. Some of them are not particularly cheap. Sometimes it's the communication between the actual scientists and the doctors, uh, and largely it's also the availability of the test. So if we spoke about this before as well as okay, I read this paper here in South Africa that was published in this European journal, and it clearly found okay, so psychiatric cases are often misdiagnosed as a psychiatric case when actually that's just a symptom of a rare genetic disorder. And when you do rare genetic disorder tests, you find out what the actual thing is. Now, given that I've told you that, if you're one of your patients presents with psychiatric symptoms along with comorbidities of other genetic, possibly genetic regulatory things, where do you go? Where do you send them? What test do you prescribe? I don't know of any company that does rare genetic testing in South Africa. Um,
I think that's the real point of this whole discussion, is that
yeah,
there's a urgent need for accessible genetic testing, but focused as well.
yeah,
And looking at rolling this out into clinically oriented testing with good education and guidance on how to use it.
yeah.
And one would definitely need buy-in from the organizations and the different regulatory groups, HPCSA, and ultimately,
Safra.
yeah, the national NHLS might need to be able to get involved for government-based testing, because I mean this could save the government a lot of money. Psychiatric hospitals are often full, oversubscribed in terms of patient load. And if you can get these people treated in other psychiatric hospitals, there will also be a huge cost saving in it. So, um,
Absolutely.
yeah, there's a lot to do in this whole thing. If you had to pick one sort of way of rolling this out as a genetic testing service, what would you do, and how would you sort of target it?
Yeah, so that's a good question, and it's always the question that comes up with rare genetic diseases. So I think it look, a lot of these things have atypical progressive treatment resistance or multisystemic symptoms. So when you in another way of phrasing it, if you see two types of symptoms or a list of symptoms that don't fit a box of a specific disorder that you're used to seeing at all. So think of a clinical example. These are almost always the case symptoms that you see with this, but now you have this patient that they have all of those, and then they have this almost strange extra symptom. Then that would be a good funnel to sort of start thinking, okay, perhaps we should do a rare genetic test. But in terms of rolling it out, so from the company actually doing these tests, is you it wouldn't make sense to have a assay, because if you have a whole assay, that you will always only be testing for, it's already unlikely to have one rare genetic disorder. It's incredibly unlikely to have two different rare genetic disorders. So it doesn't help testing for a panel of possibilities. That's not the most that's financially not a smart move to make. So you would have to do it sort of PCR based, where you have your primers for specific known variants that are rare genetic based, or you would have to do whole genome sequencing if they're polygenic or regulatory based or microRNA or epigenetic based. And the big problem with that is is that to do a whole exome or whole genome sequence, even if you do it cheaply in relative terms using something like the Illumina platform, it'll cost you about 11,000 Rand just to run that thing once. And that's if you have 64, I think I'm under correction, I think it takes 64 patients at or samples at a time. But I'm actually not 100% sure. But that's given a full plate. So that's whatever the max capacity of patients is. So that means you have, let's say it's 64. That means there are 64 people, the demand for 64 people who have some rare disease, that you're running through this whole genome sequence once to make it financially available. And so it's difficult. And that machine costs, just to maintain every year, a few million Rand, and to buy something like 5 million Rand or 10 million Rand or something like that. So it's an incredibly expensive piece of equipment to do. And so I think the biggest hurdle to companies doing this is it's too risky. Even though it's clear that this will help people and that this can improve clinic from a financial perspective, it's very hard to commercialize that and actually make a big profit out of that. Um, yeah, so that's what I think is what's holding companies back from going full into that, is because they will likely lose money.
Yeah. But do you think the rare diseases diagnostics is the real holy grail, or do you think it's more really getting to understand the genetics of the more commonly admitted and treated psychiatric disorders?
Well, that's such a great question. And so that's exactly it. So yeah, the Holy Grail, it's not rare diseases, but if you're doing a whole genome sequence, that you hold that for your whole life. That's a lifelong result that you have. And so any time for the rest of your life when you want medication prescribed, whether it's neuropsychiatric or oncology based or it doesn't matter what the consequent test is, you will always have your genes available. And in that way, it does it does make sense financially. 11,000 rand once off in your life, and then you have the potential costs of what you can save in terms of misdiagnoses and wrong prescriptions and that type of thing
Hmm.
is immense for the patient, for the client, to in the business perspective. Um, so it does absolutely make sense to do this, and
Hmm.
especially it makes sense to get life insurance and medical aid company, not life insurance, sorry, medical aid companies involved in this type of thing because it not not in terms of them having the data. It's a very important thing to just really emphasize is we can't the data security of having people's genomes is a whole other ethical issue, but getting medical aids to support financially to get make it standard practice to get genomes sequenced so that we can uh, have a better idea of what to prescribe people as they go through life and diseases and disorders start presenting. Because right now there are so many cases of misdiagnoses or like in that example of the 2024 study where there was the right diagnosis that is the disorder that they have, but it's not the right medication or it's not the right amount of that medication, or it didn't consider that this is a genetic variable and your family also has this. Or there's a you don't you have this, no one else in your family has it, but your brother is very likely to give it to his children, and that may be something that you would want to know, that type of thing. And that's all the information that you would get with whole exome or whole genome sequencing. Whole exome sequencing, just to clarify what that term is. So that's looking beyond just the genes. That's actually looking at how they're expressed. So that would be looking at things like microRNA levels and methylation patterns and that type of thing. You get different variants of it, but it's looking not just at the code, but at the expression of the code as well, which is something that can change throughout your life, which is the topic of epigenetics, how the environment can affect these things and sort of dampen genes and make them enrich genes, make them express more and that type of thing. Sorry, Stephen, you're muted.
Sorry, because I unfortunately have an emergency in the hospital, so I'm gonna have to leave. I'm so sorry about that.
Okay,
I'm just being
no
called.
problem. Yeah, no,
But yeah, but it's great. I think we should continue a psychiatric session at some stage, but I'm sure you can continue. I'm sorry about that.
absolutely.
Thank you
No
very much.
problem. No
Thank
problem.
you.
Cheers.
Okay.
Thank you for joining.
Thank you. Bye.
Yeah. So let me continue actually. So there, apart from the study on the rare genetic diseases and the gene screenings and that type of thing, there was also a study done uh, looking at neuroanatomy. So that's of course the other side. If you're talking about neuropsychiatric genetics, you're not just talking necessarily about drug pathways and circuits, but you're also looking at the anatomical correlation. Or let me phrase that in another way. So when you're looking at cofactors and covariables, you're not just looking at these genes and their risk profiles and how that associates with drugs, but you're also able sometimes to physiologically look at how does this change the brain structure, right? So there was one study specifically that looked at the striatum. Um, so this is in the brain, in brain terms, this is the caudate and the putamen. And essentially what this whole system does is it connects the cortex in the brain to the basal ganglia, to the thalamus, and then back to the cortex. So for the clinicians, what this essentially does, this part of the brain does, it's part of the reward system, the motivation system, habit formation, cognitive control, and sort of action selection. And in terms of disorders or diseases, it's relevant to depression and bipolar disorder, and to some extent schizophrenia as well. So you can start thinking neurochemically. This would mean that there's probably things like norepinephrine and dopamine and serotonin and these neurochemicals involved as well. Anyway, so the striatum is a structure that's organized in a way that mirrors brain development gradients, gene expression patterns, and psychiatric genetics, or psychiatric gene risk. And so, in this study that I started talking about now, what they did is they looked at the microstructural covariance between cortical networks and striatal subregions. Well, that's a lot. I'll go into the terms in a second. And they used as something very similar to MRI. So they use it's called T1W T2W mapping. It's basically MRI, but it also looks at, or it can also take into account myelin sheaths. Um, and then so they took this brain image, the MRI similar thing, and they looked at how does that correlate, the image correlate to gene expression profiles and psychiatric risk genes. So, okay, just to define a few of those concepts quickly. So the microstructure refers to tissues that feature like myelin, myelin sheaths or cell composition. Um, so it's the material properties of what you're looking at. That's the microstructure. Covariance in this specific case would refer to two regions that vary together, so suggesting some sort of linked organization or shared developmental constraints or underlying factor that influences both. And then the gradient is, in the case of the brain striatum, is a smooth change along the axis from the anterior to the posterior. So basically, what they found in this study is that there was a microstructural gradient along the anterior to posterior axis of the striatum. So there was a change in smoothness from the back of the brain to the front of the brain, and and that they used this thing similar to mRNA to measure this. And what they saw was that the anterior striatum is more enriched in psychiatric disorders, right? And so what they derive from this is that they can relate the covariance patterns of cell type specific gene expression, and they could see that there was a positive correlation between gene expression profiles and excitatory or inhibitory neurons, and a negatively associated correlation between gene expression and astrocytes and microglia. It's a lot of terms, but essentially different parts of the striatum align with different cellular biology and cellular biological signatures. So this matters because psychiatric risk is often about which cell type and circuit and pathway are most vulnerable, and not just the diagnosis label. So when we talk about enrichment in psychiatric risk genes, what is enrichment referring to? So here it means a sort of overlap between the expected, or an overlap that's more than expected between the gene and the risk. So if you see a certain correlation between risk genes being prevalent and a certain anatomical structure, and you see that these two occur together more commonly than that would be expected based on statistics, then you can see, okay, these are enriched. So, and the specific study did report on these enriched signals, where they did the multiple comparison correction. So, this study was also specifically on schizophrenia and bipolar, depression, and those type of disorders. And so, essentially, what they were seeing there is, okay, brain regions, anatomical structures, gene variants, gene expression are all linked in a network-based system, a circuit-based system. And actually went further on in a follow-up study to validate that as well. So they used so in the first study where they used something similar to a MRI, and the second study they used something similar to a fMRI, functional magnetic resonance imaging. So the thing that they used is called a support vector machine, or SVM classification. And essentially what they found is that there was accuracy in when they associated these brain regions to the different disorders, specifically for the disorders of depression, schizophrenia, bipolar disorder. And so to translate all of that simply, apologies, I know it was a lot. So this suggests that circuits that they identify from transcriptomics, so this is this whole sort of exome sequencing thing, and the microarchitecture aren't just theoretical. They carry a physical functional data where you can distinguish from patient groups with controls with various strengths anatomically. There are differences between these patients, and you can physically see this with the physiological expression of the brain development or the brain expression of neurochemicals and that type of thing. So, what does this mean practically in the clinic? So, they, I don't think anytime soon there won't be a striatum gradient test that a doctor can order, but it does support the clinician mental model that neuropsychiatric illnesses is partly a circuit or a developmental problem. So where genetic risk is expressed as altered development organization, cell type vulnerabilities, and changes in specific certain pathways. In that specific case, it was the medial frontal limbic visual striatum can be linked to different psychiatric disorders. So brain anatomy is another thing that's also involved, is the take-home message. Uh, but anyway, back to the clinical reality of genetics and how it's integrated into psychiatry. We spoke a little bit with Stephen about this caring clinic where they physically took the the results of trying to implement genetic care or genetic screening into the test and seeing how that affects diagnosis. And just to recap what I told Stephen as well, what they found is not necessarily better diagnosis, but it did change or did influence whether they did cascade testing. So family testing is another word for cascade testing, whether that involved family counseling, medical changes, clinical trial referrals, additional medical surveillances, specialty referrals, and medication adjustments. And then the follow-up study to that, just to recap again, was a rare disease study where they found a lot of psychiatric or a lot of psychiatric expressions, which was misdiagnosed as a psychiatric disorder, was actually just a symptom of a completely different rare genetic disorder. So, to summarize all of the things we spoke about today, is how do you explain neuropsychiatric genetics to patients? So, in mental health genetics, usually you know that you know now it doesn't act like a on or off switch. It can in monogenic variants, but for the most part, it's a dial. It's a regulation thing. It's how much, when, where. You also know that your genes can influence how the brain development develops and how brain circuits regulate mood or attention or motivation or thinking. That was that whole last bit that we spoke about how there's functional neuro and anatomical changes that can be observed sometimes, or neurodevelopmental changes that can be observed. We also spoke about how the environment and life experiences matter. I spoke about childhood trauma being an influencing factor and how these things can, while they don't necessarily change the underlying genetics, they can change the expression of those genes through something like microRNA. And then we also spoke about understanding genetics and how that can sometimes change monitoring of drugs or family counseling or occasionally even the treatment, so even changing to a different treatment. Just to hook that into biology, so a lot of things change the risk of regulation. Things like microRNA show strongly how different levels of microRNA, we spoke about the QTLs, how that can directly also link to how different genes are expressed in specific types of brain tissues. We spoke about the very first study that was done in the prefrontal cortex. So it's the same genes, but localized in that area of the brain. Those same genes are being expressed differently because they're being regulated epigenetically by something like microRNA, and that has an effect on gene diagnosis, disease diagnosis, and yeah, so that is an important take-home message, is that not only it's not just your genes which are the same in your nucleus everywhere in your body, but it's per specific localized cell area, like in the brain and neuropsychiatric disorders. You can have the case where in that specific tissue, it's being expressed differently, right? And this is where the field of epigenetics becomes really complex because if you just do a regular genetic test, say from cell-free DNA or from drawing blood or something like that, you won't necessarily see any changes. So that's why you have to, before you can just say correlation, you have to include multivariables from functional magnetic imaging to statistical models to looking at the biochemical pathways. Neuropsychiatric genetics, at the end of the day, is a very complex and multi-field study. Actually, it's not if you can't just get and do genetics and have the answer. You can't just do neuroscience and have the answer. It does, unfortunately, not work as simply as that. You do need to take into account many different factors. So let me think about some take home points out of all of that. So the neuropsychiatric genetics, mostly the genetics part of it, mostly refers to risk architecture. So you can see the risk of, or you can see genetically that this gene is producing more of what more microRNAs, and therefore its regulation is affected. Let's say the regulation is affected that the microRNA silences the messenger RNA. So this gene in this specific tissue is not expressing as much of a protein as it should, or at least in theory, that's what we can see from the genetics. And that would be a case of this person is at a higher risk because in a case where this gene is not being expressed as much as it should, we found when we linked it to certain diseases that that's also associated with a risk or with people who have that disease. So it's all this cascade of correlation. Uh, it's also important to realize that genetic expresses itself through cell types, different cell tissues, different locations, and different circuits. So we spoke a little bit about the corticostriatal organization, um, and that's the one with the gradient change from the posterior to the anterior side of the brain in the striatum. Another big important take-home message is integrating genetics into care can change the management pathways of neuropsychiatric symptoms and can sometimes precede rare diseases. So that's the case where you see neuropsychiatric disorders and you want to diagnose it as a neuropsychiatric disorder, but you also see other symptoms that are not typically associated with that diagnosis. So instead, then, of saying it's this disorder plus a few extra symptoms, you need to consider the fact that this might be actually underlying to a completely different thing. This might be underlying to a rare genetic disorder. So if you remember nothing else, genetics gives us a deeper systemic explanation of why neuropsychiatric illnesses look the way they do, and that can ultimately improve care when it's integrated thoughtfully and ethically. And with that, I'm going to leave you for tonight. And thank you very much for all the participants that joined tonight. I hope you enjoyed it. You're welcome to email me or send messages and ask questions if you have any further question on this type of thing. And I look forward to seeing all of you next week. Cheerio, good night.